Behind the Kavli Prize are three partners. It is the Norwegian Academy of Science and Letters, which I represent, and it is the Kavli Foundation in California, and it is the Norwegian Ministry of Education and Research. So why this occupation? Well, first of all, I will show a picture demonstrating, proving that Rothman received the Kavli Prize in 2010. He is in the middle here, receiving it from His Royal Highness, the King of Norway, together with the other two in neurosciences. Now, why this preoccupation with Norway in the Kavli Prizes? The reason is this person, Fred Kavli, who was born in Norway. He was a physicist and engineer, educated in Norway, moved to the US and became a very successful developer of sensors to high-tech applications. And he left behind a foundation which is going to philanthropic purposes. So that is the reason why there are two Norwegian partners. Another picture to show the Norwegian flavor here. This is all the Kavli Prize winners in 2010 in the so-called Edvard Munch room in the Oslo City Hall. And here we see, together with the prize winners, we have Fred Kavli in the back there, and we have the King of Norway, and the then Minister of Education and Research in her costume. And we have tonight's speaker to the right in the picture. And then I will, as the representative of Kavli Foundation, I will introduce Eric Marshall.
Well, good evening, and I'm so happy that all of you are here tonight to join us. First, I'd like to thank Dr. Matthew Scott, and also you'll meet Mr. Marius Deerdahl and Dr. Øystein Hove for co-hosting this Kavli Prize Laureate lecture tonight. So since the Kavli Prize was first awarded in 2008, we've held an annual Kavli Prize Laureate lecture here at the Carnegie Institution. And we're so pleased this year that we have Dr. James Rothman here to share the evening with us. And following his lecture, we have a treat in that Mr. Frank Sesno, who's a former CNN correspondent, anchor, and Washington bureau chief, will be interviewing Dr. Rothman and sharing some questions and answers with us. So we're excited to hear about Dr. Rothman's work in discovering the molecular basis of neurotransmitter release. And as mentioned earlier, the Kavli Prizes are a partnership between the Kavli, or between the Norwegian Academy of Science and Letters, the Norwegian Ministry of Education and Research, and the Kavli Foundation. And the Norwegian Academy of Sciences and Letters is explicitly responsible for the nomination process, the selection process, and announcing the prize. Now, the Kavli Prize is just one of many programs of the foundation. Fred Kavli, whose picture you saw, who passed about 4 years ago, was, as was mentioned, a Norwegian-born American who had a desire to support basic science research. And in the year 2000, he founded the Kavli Foundation. Now, the Kavli Foundation's mission is very similar to that of the Kavli Prize. The mission is dedicated to the advancement of science for the benefit of humanity, the promotion of public understanding of scientific research, and also to the support of scientists and their work. To quote Fred Kavli, "We support the dreams, the ideas, the ambitions, and the hopes that we will find things that are revolutionary in the future." And the vision and mission of the Kavli Foundation is implemented through a series of programs, including the prize itself. We have 20 Kavli institutes at major research institutions around the world, and we hold a variety of Kavli symposia and meetings to help science move forward. So the Kavli Foundation is deeply appreciative to our founder's vision and his philanthropic impulse to provide funding for science. Fred Kavli's legacy will continue into perpetuity. and support basic science, and also to enhance the public understanding of scientific research through events like this one here tonight. So I thank you all very much for joining us. And now President Matthew Scott will provide an introduction to Dr. Rothman.
Okay, thank you, Eric. A human body has tens of trillions of cells, all derived from a fertilized egg, which is a single cell. Within each cell is a remarkably complex machinery that controls the shape of the cell, if and when it divides into two daughter cells, the production of new molecules, and the locations of the cell's parts. The interior of the cell is a very busy place with lots of movement of molecules and of small bodies called organelles. The movement, sometimes called trafficking, unlike Washington traffic, which doesn't move, must ensure that different parts of the cell machinery are sorted out and delivered to different locations. You need the bricks in the right place to build the wall and the pipes in the kitchen to plumb the sink. For example, in neurons, small organelles, membrane-covered, fluid-filled bags of molecules called synaptic vesicles, are transported to an area near the surface of the cell in one part of it in order to communicate with adjacent neurons. That's how we think. That's how I'm talking. That's how you're listening. There are many kinds of organelle, each with a specific function, and each has to get to the right place. How do the movements happen? What are the conveyor belts? How does the cell's GPS work? And where are the barcodes? How do movements change as the cell responds to its environment? Understanding how cells orchestrate their internal traffic has been a major goal in biology for over a century. In the late 1970s, our guest tonight, Dr. James Rothman, became intrigued by the trafficking challenges, what he and others called the sorting problem. It was hypothesized by then that cells transport many of their products in a variety of kinds of vesicles, including in neurons synaptic vesicles, but no one knew exactly how vesicles are sorted out and delivered to the right destinations in a busy cell. Within a few decades, Dr. Rothman and other scientists working on the problem independently and with different approaches had made tremendous progress. It was fundamental work, and for it, Dr. Rothman, along with Randy Schekman and Tom Sudoff, won a 2013 Nobel Prize. The announcement said, quote, "Through their discoveries, Rothman, Schekman, and Sudoff have revealed the exquisitely precise control system for the transport and delivery of cellular cargo. Disturbances in this system have deleterious effects and contribute to conditions such as neurological diseases, diabetes, and immunological disorders," unquote. Dr. Rothman is the Sterling Professor and Chair of the Department of Cell Biology at the Yale University School of Medicine. He is the founder of Yale's Nanobiology Institute. He attended Yale as an undergraduate and earned his PhD at Harvard. After a postdoctoral fellowship with Professor Harvey Lodish at MIT, he then had faculty positions at Stanford, Princeton, the Memorial Sloan Kettering Cancer Center, and Columbia. He returned to Yale as a professor in 2008. In addition to his seminal discoveries in intracellular transport and trafficking, Dr. Rothman has made contributions to the fields of neural networks and protein folding. He's been elected to the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. He has received many prizes in addition to his Nobel Prize, including the Kavli Prize, of course, we're celebrating tonight, the E.B. Wilson Medal, the Louisa Gross Horwitz Prize, and the Lasker Award. All are testament to the importance of the cellular processes his work helped bring to light. The Carnegie Institution is pleased to join the Kavli Foundation, the Royal Embassy of Norway, the Norwegian Academy of Science and Letters, and the Norwegian Ministry of Education and Research in welcoming this remarkable scientist. The title of his talk tonight is right there, so I won't read it again. Ladies and gentlemen, Dr. James Rothman.
Very good. Can everybody hear me okay? Okay, terrific. Well, thank you. I'd like to thank all of our hosts for welcoming us tonight. I must say there are so many hosts. I was a little concerned that we might not have enough guests, but I can see that actually there are a lot of guests, and I especially want to thank all of you for coming in an evening, especially such a nice evening as this, and spending it indoors. In compensation, though, we do have this extraordinary auditorium that I, for one, haven't seen before, but it's really an inspiration. My only regret, actually, in coming tonight is that I won't be here to hear the other lectures in this series. I was looking at the poster, and I have to say the topics are really extraordinarily interesting, and it's really wonderful that the Washington area has such a vibrant scientific. Why don't we really just get right into it? With that wonderful introduction from President Scott, I almost don't need to give an introduction, and therefore I won't because my time is very limited. But what I would like to say is the delivery system that we heard about that consists of what we call vesicles, and he mentioned the term synaptic vesicles. So what's a vesicle? A vesicle is a little spherical packet. I'll show you some electron micrographs in a moment. It's very, very small. It exists on the nanoscale, so many, many thousands of them can fit even within a small part of one cell. So on the nanoscale. So how small is that? So if you take 500 hydrogen atoms and line them up in a line, that's the diameter of one of these typical vesicles. That's how small they are. Small should not be confused with lack of importance. Okay? Now inside, even though that nanospace is very small, they pack a lot of punch. And inside of one of those vesicles at a nerve ending, you might have 10,000 neurotransmitter molecules. Inside of one of the packets that Dr. Scott mentioned that move proteins within the cell, you might have several hundred different working parts of the cell called proteins. So these are very potent machines. Now, within the cell, you have, as I said, thousands of these, and each one needs to know to go to the right place to deliver its content. So some of them hang out and concentrate underneath the surface of the cell. For example, the ones that release the neurotransmitter concentrate at a nerve ending, and they release the neurotransmitter to pass an electrical signal through chemistry from one nerve to another, through neurotransmitter chemistry. Other types of vesicles, perhaps in different cells, also are underneath the membrane, but they have different stuff inside. They have different cargo. For example, insulin. I think you know a lot of the cargo. The insulin that gets released to your blood after a meal, which you've not yet had, but if you had it, would be, or perhaps you have had it. And that insulin is released by these packets. But most of the packets are actually held within the cell, and they are formed in one location, they traffic to another, as you heard, and they deliver their cargo. The amazing thing is that in this sea of vesicles, each one has to find the right place. If they didn't, then the organization, the very precise organization within each of our cells, would be lost in a matter of minutes, because a vesicle is born as it buds from one location in the cell, and it disappears, it is consumed as it delivers its cargo by membrane fusion to another. It might live on the average only a minute or two. And so the flux of these vesicles, some are long-lived, but some are not, is extraordinary. And the variety of these vesicles is equally extraordinary. And this happens to be a human pancreatic cell. This is the Golgi apparatus, the so-called endoplasmic reticulum. And here, can you see these tiny little vesicles? Smaller than all the other organelles. These particular ones are carrying newly made proteins from here, where they were just made by ribosomes, to here, where they will be modified in the Golgi apparatus to be prepared generally for release from the cell. And there are perhaps, oops, I'm going backwards. This is the danger of Fred Kavli. You just can't get away from him. And actually, everywhere I go, there's Fred Kavli. But actually, he was a great man, and he continues to contribute through his foundation. So here you have perhaps 20 different types of vesicles that mediate key aspects of cell growth and division. There are dozens of different types of vesicles that focus on the cell surface to release hormones. And the same types of vesicles store the receptors that bind these hormones. And believe it or not, about 20% of our genome is devoted to the kinds of products that are carried in these vesicles. And probably the most evolved vesicle of all is at the nerve ending, the end of every nerve. Here's the synaptic vesicle that you heard referred to before, which on command of the nervous impulse releases the neurotransmitter by this process called membrane fusion. Now, remarkably enough, the very same process, this happens to be storing digestive enzymes, this so-called secretory vesicle, it underlies the plasma membrane. It will release its content to the outside of the cell, which in this case means the GI tract, and it will go into your intestinal tract. And the same underlying machinery, and this is what I'm going to tell you tonight, my message as it were, is that there's a single protein machinery called snare proteins that we discovered that is responsible for all of these diverse physiologically important events, whether you're talking about a human cell, a cell from the simplest eukaryotic microbe like a yeast, or any plant cell. It's a universal law that applies. So how did we discover this? Well, any discovery relies on another discovery. In the field that I work in, the field of cell biology, was founded by George Palade, who discovered these vesicles. He discovered the fact that there's trafficking, and he discovered through the electron microscope most of the organelles that we recognize today in the cell. And he was awarded, along with others, the Nobel Prize in Medicine in 1974. I was a student in those days, and I learned about his studies actually when I was a medical student. I'm a medical school dropout. But in the interim, before I dropped out, I learned all about histology and anatomy, and then you learn about Palade because he discovered what I described a moment ago. But what was not possible for scientists of his generation to discover, because there were no tools, was to answer the underlying question, what is it actually that allows a vesicle to form? How does that vesicle actually know where to go? You could see them in the electron microscope, you could make guesses, but how did it actually work? The first and important step was taken by my colleague, whose name has already been mentioned, Randy Sheckman. And he discovered that the simple, humble microbe yeast has the same secretory and other transport pathways that our more evolved cells have. He showed that Palade's process and pathway exists in yeast. And most importantly, he discovered a large number of genes that when each one is mutated, it blocks a specific step in this pathway. And what that meant, because a gene encodes a protein, is that there must be protein machinery underlying the process that forms a vesicle and instructs the vesicle where to go and tells it how to fuse when it reaches its target. There must be, in other words, this problem can be rationally solved by biochemistry because proteins are the domain of the biochemist. Proteins are enzymes. Enzymes mediate pathways. Pathways means chemistry. And so this meant that a biochemist such as myself could perhaps discover the enzymes and machinery that are responsible and then understand the process in chemical terms. Now, actually, that's the beginning of my story. Now, this is me, actually, at the age of 12 with a rocket. Now, why as a 12-year-old in 1962 with a rocket? Because I grew up in the era of physics. I grew up in an era, you recognize that person? Do you recognize that person? Okay, that's Oppenheimer. This is the famous picture, a famous picture of them at the Institute for Advanced Studies. That is one thing that came of that, and we were all aware of it. This is the other thing that came of it. Anybody know who that was? Yes, Alan Shepard. Most people reflexively say John Glenn, but it's actually Alan Shepard. That's in that era. The physicists actually, for those of you who are old enough to remember, were actually the most admired individuals, number one on the list. So if you like math, you became a physicist. I went to Yale. I was a physicist, a theoretical physicist, until I became a biologist when I went to medical school. So I still look at the world as a physicist, even in my current work, even though I do biology. How does a physicist look at the world? And it's this perspective, actually, that allowed me to make the contribution that I made. A physicist always looks for universal laws that explain something on a common basis, and they better be the simplest. Newton, the apple drops. And therefore, of course, the moon must obey the same laws, right? Oh, come on, okay? That was his great discovery, universality. He was the first physicist, okay? So when you think about everything that you've heard about tonight, there's actually two hypotheses. There's one that every physicist would naturally come up with, and which I came up with, and there's the other which all of my colleagues came up with, because they were biologists. These are the colleagues who all thought I was wrong. And actually, I have been wrong a lot, okay? But about one thing I was right. Anatomy dictates specificity. The field that I described to you came from the field of anatomy. You see cells, they're beautifully organized. Within the cell, there are vesicles. The vesicles going from this location, the endoplasmic reticulum, to that location, the Golgi, are beautifully sandwiched in the two, between the two in the cell. So it's not hard to imagine, as everybody did, frankly, that this vesicle didn't need to know where to go. There was nothing special about it. It was simply born here, and it died there when it fused, and there was no place else for it to go because it was confined. Anatomy dictates specificity. Now, the other is to say this vesicle actually carries information within its own protein composition that says, I came from here. And that protein has a pilot, and the pilot guides it to a receptor. And those two proteins interact and then trigger the fusion process. This is actually how a physicist would think about it, because the specificity dictates the anatomy. And let me illustrate that to you in the following way. Imagine a vesicle is an apple, and the organelle where it comes from, the endoplasmic reticulum, is my hand. And I want to get it to the Golgi. I want the apple to get into my mouth. Okay? Well, anatomy dictates specificity is the following. The two are near each other. I just put it in my mouth. The apple is stupid. There's no information at all. But this model says I throw the apple up in the air, I merely open my mouth, and it finds me. Okay? Now, according to this hypothesis, which rejected the idea of any intracellular organization being of any importance, no matter how beautiful it was, there was a very simple hypothesis. The hypothesis is that vesicle traffic, an example of the very process that generates the organization that we admire in the cell, can nonetheless take place when there is no existing cellular organization. And if that's true, that hypothesis can be tested. And in 1980, we tested that hypothesis based on someone who actually invented that whole way of thinking. Nothing ever comes from, everything comes from something. The founder of biochemistry, the essence of biochemistry, is this hypothesis, is Edward Buchner, who received the Nobel Prize in 1907, the first biological Nobel Prize in chemistry, actually. And it was for discovering that fermentation, alcohol formation by yeast, can occur in a cell-free extract, thereby proving that actually Pasteur, the great scientist of the time, was wrong in believing that there's something special in a living cell that's something other than chemistry. And he wrote in 1907 that we are seeing cells more and more clearly as chemical factories. In other words, there is nothing special about life other than the fact that it is the amazing result of many chemical reactions. Well, the entire field of chemistry and molecular biology has been built on elaborating that idea, and my colleagues and I, initially at Stanford, built on it. And what we did is very simply shown here. We said, here are two organelles, it doesn't matter the technical terms, A and B. And we have a vesicle that goes between them. And normally, within a single cell, that vesicle will go from A to B, somehow. Now, what if we actually broke that cell open and added to it the same compartments, A and B, from a completely different cell? And what if we could observe that our cargo got packaged into a vesicle and it went from here in one cell to there in what was inside another cell? And what if that happened efficiently in the test tube? Well, then it must mean that this vesicle was able to go on its own, just like that apple, and find its way by diffusion. And it must mean that it has built-in specificity, and the basic hypothesis is correct. Well, that's exactly what we found in 1980. Okay? And what it enabled us to do, because this only happened when we added the cellular energy source, ATP, and only when we added the cytosol, or the sap fraction of the cell that contains perhaps 10 or 20,000 different enzymes. And our goal then became to discover which ones were actually making the vesicle and which ones were directing the vesicle. And we did succeed in that. It took many years, but we were following in the footsteps of great biochemists that enabled us to do that. Now, people always ask me, what is your advice for a young scientist? Now, actually, I was a young scientist when this happened. I was 30 years old. And I have to say the first thing that happened is my colleagues questioned the veracity of my observations. Not that I was reporting fake experiments, but how to interpret them. And they were right to do that. And as a result of those challenges, I upped my game, and over many years was forced into a level of scientific proof which ultimately was very satisfying to everyone. But I would point out a quote here for any young scientist who does something original, because when you do, actually, you don't know how original it really is, and it may be wrong. But here's Kepler, okay, who, when he wrote his great work, Astronomia Nova, 1609, wrote the following in the introduction: advice for idiots. But whoever is too stupid to understand astronomical science, meaning doesn't believe in what he's writing, or too weak to believe Copernicus without affecting his faith, thinking of Galileo, who was his contemporary, I would advise him that having dismissed astronomical studies and having damned whatever philosophical studies he pleases, he mind his own business and betake himself to scratch in his own dirt patch. Now, I know, I thought that, but if I'd have said that, that would have been the end of my NIH funding. Happily, I was able to restrain myself, and with many colleagues over the years, we learned how to produce the vesicles. So these are the vesicles that carry my cargo protein in the process that we reconstituted. And what's new about them is they're not being seen in the electron microscope in a cell. We produced them in the test tube from pure proteins that we discovered along the way, and we purified the vesicles, and the cargo is in them, and there's nothing here but these vesicles. So we really were able to say that we could understand a vesicle because we could synthesize it in the test tube from pure proteins. And as a result, we discovered the process by which these vesicles form. We could take these vesicles and add them to the target membranes, again in the test tube, and they efficiently fused. And they fused only with the target proteins. And that enabled us to discover the machinery that mediates their fusion. And that machinery is the same throughout nature, and it consists of two proteins. We call them snare proteins. A vesicle snare protein, this is the pilot protein that we hypothesized in 1982, and the receptor protein that we also hypothesized in the target membrane called the T-snare, V-snare and T-snare. And they partner each other hand in glove. And each organelle has its own version of a T-snare, each vesicle its own version of a V-snare. And as we later discovered, they partner each other in a specific way, and that tells the vesicle where to go. And once it's there, it turns out, and how beautiful is this, that the pilot proteins become the fusion machinery. They are one and the same. And you see this pin-like structure? It literally sucks the vesicle into the target membrane and it fuses it. And I can see I'm running out of time, so I'm not going to go through the details of this. But these proteins actually fold up or zipper on each other as they partner each other. So as they target, as the vesicle homes in and targets, literally these proteins fold, and as they fold, they produce an inner directed force that forces the membrane together, just like as if you were to force two soap bubbles together. So I want to illustrate the principle here. What you're looking at is the process of membrane fusion. Imagine each of these balls is a vesicle, and you want to fuse them together. And in this analogy, the snare complex, or pin, is like a hairpin. Now, you've all had the experience of opening a hairpin. What happens? It takes energy, work, to open the hairpin, right? If you let it go, what does it do? It snaps back. Now, in your mind's eye, imagine doing that to the hairpin, and now inserting each end into one of these balls like a vesicle, okay? And now letting go, and that will happen again on the next cycle. And what will happen? The pin closes, it's the inner directed force, and it forces the two together. Now, why does it force it together? Because these membranes are like liquid, they're deformable. They're made of fatty lipids that are deformable, okay? If these were tennis balls or golf balls, they would just stick together, and they would be forced together, but because the membranes are compliant, they fuse. So that actually is how membrane fusion works. And I would be remiss without showing you a single experiment, so I will only show you one, done with a colleague recently, now 5 years ago, at Yale, Yongli Sung, a professor in my department at Yale. And just so that you believe in the force, I don't mean that to sound like Star Wars, but here what we've done is we've taken a single snare complex, the V and the T snare that have assembled together, a single molecule, and using some special tricks, put it between two glass beads. Then we use two laser beams, starting to sound like Star Wars, and we put one on one glass bead and one on the other, and the laser actually is like a tractor beam for those glass beads, and then we can move them a couple of angstroms at a time. And as we move them apart, we can actually exert force, and as we do, we can melt the protein, which actually reverses the fusion process. And then we can watch it, we can let it go and watch it flicker back and forth. So we can actually measure the very force on a single molecule that I've shown you that we postulated years ago to drive membrane fusion. So to summarize then, what is absolutely remarkable is that the very same protein machine in one or another iteration throughout all of biology and certainly throughout all of our cells mediates cell growth and division through the fusion of vesicles needed for the production of new components in the cell, endocrine and exocrine physiology through the provision of hormones and their receptors throughout the body, allowing cell-to-cell communication, organ-to-organ communication, and of course allowing the brain to function by allowing electrical signals to be converted to chemical signals so that electrical circuits can operate and that I can give you this speech tonight. So obviously all of this involves the work of many people and most importantly involves the scientific method. I'm often asked, what does it take to do something like this? And the answer is not much. It takes some support, it takes money, it takes an imagination, a little bit of risk-taking. It takes a lot of prior knowledge, our colleagues, and mostly it takes the scientific method. And everything that we know in science, it comes from the greatest discovery ever made in science, the only truly unique discovery, which is the scientific method itself. And as Buchner, who was, as you know, you can see a hero of mine, laid out in his own way. And I've had the privilege of working with many students and postdoctoral fellows. There are about 300 people who've contributed, each in their own way, to this project over 30 years. And so we've had a lot of fun blowing bubbles. These are little bubbles in the cell, and we've been having a lot of fun over many years illuminating what we think is an important landscape in science and medicine. So thank you very much for your attention tonight. And I particularly wanted to thank Frank Cesno, who's taken his time and energies to moderate the session that's coming next, which I'm very much looking forward to. Bravo. Thank you.
So this is a little humbling. As I was listening and watching, and this is great. You put up Alan Shepard there. And you were motivated by Alan Shepard to go and make this unbelievable discovery and win the Nobel Prize and change our fundamental understanding of how we operate.
I'm wondering if you need a microphone.
I have one.
Oh, okay.
Can you hear me?
Okay, good. Thank you.
I saw Alan Shepard, and I asked my 8th grade teacher how astronauts go to the bathroom when in weightlessness. You went and got a Nobel Prize. I became a journalist and went into fake news. So, you know, it's... So you may or may not have downloaded this app. And if you haven't, you should. Pigeonhole, and you can see, I can tell you where you need to go for this. But along the way, you can put in a question on your cell phone if you'd like. If you go there, you will see questions. And if you want to vote on those, we'll be incorporating those into our conversation here. And I can tell you that some of the questions are already running away with us. So I'm going to leave that suspense for you. I will come back to those in a few minutes. You do not seem like a dropout of any kind. Why did you drop out of medical school?
You know, I'm still trying to explain that to my mom.
She wanted a doctor, huh?
The reason is I recognized that I was never going to practice medicine. I thought there was a fair chance the patients wouldn't want me to practice medicine. But actually, I just love science, and I was really fortunate while I was still a medical student that I was offered my dream job, which was to work as an assistant professor in what was then certainly the greatest department of biochemistry at Stanford, in the shadow of the person who was my scientific hero, and I think one of the great, if not the greatest biochemist of the second half of the 20th century, Arthur Kornberg. And so there was an opportunity I couldn't pass up.
And that picture of you is what was it, 12-year-old with the rocket?
Yeah.
So you had science in your blood from when? Were you 4 and discovering things, or?
Yeah, I was pretty much a nutcase.
What was it? Where did that come from?
I don't know. It's funny, one of the things we were talking about before. I think that there are some people that have something built into them. And somehow I was always going to be a scientist. I don't know. I think part of it is the zeitgeist of the time.
Yes.
As I was mentioning, my dad was a practicing physician who I think would have liked to have been a scientist in another era. So I was always encouraged in that direction. But there was just something in me. And at one point it was a physicist. At another point it became a biologist. But it was always a scientist.
Let's talk about this for a minute.
Which, by the way, makes me very lucky, because I just want to say that not everybody's that fortunate to have a sense of direction and mission. As a parent, and all of us, I think, have had issues and challenges in that area, and you're just a very lucky person if you feel that directed, and I feel privileged for that reason.
Oh, it's amazing yes, and what's come out of it is phenomenal. Let's talk about that and what's come out of this work. Knowing, and Dr. Scott mentioned earlier this idea of disease and diabetes if the machinery breaks down. Mm-hmm. So what does this discovery, this idea of how this incredible chemical factory moves around and communicates and releases, how does that affect the way we are and think?
Yeah, okay, so, and this comes to it's a really good question, Frank, and it comes to, I think, I'll just like to generalize it a little bit first, which is, this is an example of what's called basic science. I like to use the word foundational science, actually, more than basic science, but, and so it's natural for people to say and wonder, what drug comes out of it? What medical improvement comes out of it, because after all, it was paid for ultimately by taxpayer money.
Right.
That is meant to improve human health. And there's and in this particular case, and I think it exemplifies a lot of basic or foundational science, I think of it as kind of, in other words, what I'm doing is as a result of this, and many people who engage in this activity, we create illumination. Okay. Now we don't have the privilege necessarily of seeing this translate into a new drug, let's say for Alzheimer's directly. If you can't really do both at the same time effectively, but what we're doing here is we've illuminated a whole set of concepts that in fact are enabling people to, rather more specifically, develop drug targets and pharmaceuticals that in academia and in industry that are increasingly impactful in these diseases. Mm-hmm. The value of this type of research is that it illuminates, as a holder of the lamp and a kindler of the flame, you illuminate not just many things at the same time. And so I have to admit that I used to get a little sheepish when someone would ask me the question, okay, well, so what's this good for? and actually, I think that people who do this maybe have more impact ultimately, okay? Because look, the chances are, if I focus on disease X and target Y, as hard as I try, my chances of coming up with something that matter are going to be very small. We want to have a lot of that activity in the end so we succeed, but on the other hand, my chances of impacting something probabilistically, maybe not in a determined way, but contributing to the idea that lets the next guy down the chain make that discovery that will lead to a drug is much higher.
Which I understand. If the GPS doesn't work, and diabetes was specifically mentioned, what's that connection?
Well, okay, so there are, there are, that's a complicated question to answer, but I'm going to give it a try. These are the genes that control these processes are so fundamental that if you lose that gene completely, it's simply incompatible with life. Okay? On the other hand, most of us in this room are probably walking around with mutations in the very genes that control the fusion of the vesicle, the zip code, the GPS, et cetera, et cetera. I can almost guarantee you that. Okay? So how do I rationalize those two statements? Because not all genes are complete, not every gene completely eliminates the function of the protein. So that most genes in the population have a subtle effect on an activity, and what we call these in human genetics, we call those susceptibility genes, disease susceptibility genes. And what my colleagues in human disease genetics find over and over again is that there are some families that will have, you name your favorite disease, that will run very strongly in the family. Okay? And in those rare families, there will be a single gene that they can map. Okay? But typically, for most diseases, that accounts for 5% of what we recognize clinically as the disease. The rest of it is a combination. You got some bad genes, okay? Those were likely to be the genes actually in our pathways. Mm-hmm. And in fact, more and more, we're coming up with examples of that. And I won't take the time tonight, but there are actually quite a few examples of genes that actually put a person at risk for diabetes, okay, or actually for cancer, for immunological disorders, and so on. And it's on that basis that the linkage to human disease is made. And it is likely that many of those will be drug targets in the fullness of time. It's a really good question.
I'm going to go to, because I want to incorporate this, some of the questions that we've gotten from you through Pigeonhole, because there are some terrific ones. There's a couple of top vote questions here that I'll come to later, because we just have to work our way there, I think.
Okay.
But George proposes the following question, and that is, can your studies about membrane fusion teach us anything about how viruses enter cells? That has 30 votes in the room, by the way. Wow. There is a comment to that, by the way. Somebody says, oh my God, yes, I want to know this.
Okay, well, George, you started a movement. And actually, it is a really, really good question. And I think the answer is yes. It turns out that viruses, in case, just to make sure everybody knows what the full impact of that question, a virus is a nanoscale particle. It's kind of like a vesicle. And inside that vesicle is the viral genome. It's often DNA, but not always DNA. And in and of itself, it just sits there. But it wants to replicate. It wants to divide. And in order to do that, it has to enter a cell. And when it enters the cell, it takes over the cell's machinery to make copies of itself, but now with its own instruction manual, which then had to get in. So it's kind of like a vesicle. And in fact, many of them are vesicles, and they have a membrane around them. Influenza virus, for example, or HIV are exactly that. They're kind of vesicles that exist outside the cell. So it's a really perceptive question. And then they stick to the outside of the cell, and they deliver their packet inside, just like the hormone in a packet or a neurotransmitter that gets delivered outside. It's actually the exact opposite, but the physics of it is almost exactly the same. Now, it turns out the viruses have on their outer surface a protein, like the snare protein, but it's not the SNARE protein, that recognizes a receptor on the surface and undergoes a change structurally to produce a pin that releases the vesicle, therefore the DNA, the genome of the virus, into the cell. And a number of years ago, my colleagues who work on viruses discovered the nature of these fusion proteins in the virus. They have nothing to do in terms of evolution. It's a completely different history, natural history from SNARE proteins. But structurally, they form pins. And the pins are almost identical in their physical properties. They don't look like the pins of SNARE proteins, but the physics is exactly the same. They exert a force, the force is the same, and if you try to melt them like with the optical tweezers that I showed you before, they're physically indistinguishable. So it's a perfect example of the physics of bilayer fusion. It's physics, okay? It's like two soap bubbles getting together. You know, you can push them this way, push them that way, but the physics is exactly the same. And whether it's coming from outside the cell or inside the cell, nature, you know, Darwin never falls asleep. Darwin is always at work and finds many avenues to get to the same endpoint, and he had to solve a physical problem. You know, he had to get that apple on the ground. Okay?
Okay.
Good question.
You are, you spoke about the scientific method and the importance of that. And I have a question that I wanted to ask you, because part of the scientific method is that moment of discovery. Well, I don't know if it's part of the scientific method, but we hope it's part of the process, is that moment of discovery. So I wanted to ask you this, but 28 other people do as well.
Wow.
Is tell us about your aha, your eureka moment.
You know, a lot of times scientific discoveries don't come with a eureka moment. Okay?
Say it ain't so.
I don't mean, no, it can be. They sometimes come in dribs and drabs, and then it's an accumulation of information. But even then, there is kind of an aha where you synthesize things together. And I've been fortunate to have had a couple of those in my life, just a couple. Okay? And any scientist is fortunate to have had a few, or even one. And there's just nothing like it. I mean, you know, there's a feeling of exhilaration, okay, I would say kind of responsibility in a way. There's privilege. You know, can you imagine knowing something that no human has ever known, ever, okay, ever before, okay, and you're the only person that knows it, right?
Okay, so your moment was?
That is, well, I mean, you know, okay. So
It's amazing, right? It's a little lonely, a little scary.
And you're a little, you don't really know that you're right, but if you are, you don't quite dare believe that you're right, okay? But, you know, it's egoistic. It feeds ambition, okay, to be honest, okay? But on the other hand, it's deeply humbling, okay? And what you really appreciate, especially someone who has a kind of a simple, you know, science has two kinds of people. There are people that like simplicity. They're the physics types. Then there are the people who like complexity. Oh, it's so complicated, okay? They tend to be more biological, okay? And the people who are simple, they always find simplicity, wherever they look. And the people who like the complex, they always find complexity. They're both right. Okay? There's no right or wrong. But as a simplifier, there's something deeply, almost spiritual when you see something, when you appreciate the simplicity of nature, like this question about the pen, okay? How beautiful is that, okay? Right? So that's what that moment is like, and I don't want to go through such an egoistic way, particular things in my case or whatever, but anybody that's had that experience is really deeply moved.
But
and if you're not, there's something wrong with you.
I wonder how you describe that and explain it to young scientists, to students, to people who are thinking about whether they're going to go into science or not.
Well, you know, I don't think that anybody should go into science for the hope of a eureka moment, because honestly, you're not likely to have one. Okay, I mean, you know, it happens, but it's a low probability event and it's unpredictable. So if you're going to work every day saying, "Where's my eureka moment?" you're not going to be a happy camper. Okay, and so if I could kind of morph that question slightly,
Sure.
Frank, and say to the question of what would you, you know, any senior scientist is oftentimes approached by younger scientists or would-be scientists or students, you know, what do you think, should I be a scientist, what have you. And, I just think that don't do it because you want a Eureka moment, okay? There's, easier ways in life to get one, okay, of one sort or another. But if you have to ask the question about whether you want to be a scientist, it might be that you don't want to be a scientist. Because I tend to think, and I've said this many times, but I'll say it again, I tend to think of science as sort of an art. And there are some scientists, like artists, if you were, if a young novelist says, I'm going to be a novelist, and you say, well, that's, think to yourself, oh man, that poor guy's never going to earn a living. Okay, but but that's not fair, because that person, that is an artist. He can't do anything other than that. That's what he was born to do. And if he didn't or she didn't do that, it would be like their soul was ripped out of them. Well, that's how I felt as a young scientist. And I think that the real reward, okay, is not that Eureka moment. The reward is to be able to practice an art. Okay? And it's an amazing thing that we live in a society that, at least until recently, that capability hasn't been, really under threat.
Well, which takes me to a question that has 34 votes. And I would ask you to answer this as a scientist or a physicist, if you can. It's not a very scientific question, but it certainly is interesting. What do you think is inside of Donald Trump's brain? I'm merely passing it along.
I think I'll pass on that.
Thank you. I was actually hoping you'd do that. I want to thank my friend Nina Fedorov here, because in a book that I was working on, I wrote a chapter on scientific questioning.
Yeah.
And in talking about the moment that we're in this country and thinking about science, I really believe that this may be a part of the fundamental problem. One of the most important parts of scientific questioning and the scientific method is the understanding, appreciation, and embrace of this notion of uncertainty. But we live on a planet where people want to be certain about things. We want definitive answers. How do you, as you're talking to people in different walks of life, how do you explain how scientific uncertainty is something that people who want certainty can put their arms around?
Well, that's interesting. So, you're referring probably in some way to climate change and that sort of thing, right? But, in the laboratory sciences, the hard sciences, I think, are inherently very different than that, in the sense that you obtain information in completely controllable contexts, where all variables are controlled. And really the art of science is the capability, the learned capability of being able to create situations that are artificial, in which a concept or a principle can be tested unequivocally. And that's really actually at the level of a mathematical proof. And it's often said that in science you can't prove something, and if that's true, and that you can only disprove it. But I think, and that's literally true unless you can control all of the variables. Now, to the extent in practicing science in the real world, we oftentimes can't control all the variables, but we can come very close. And whereas, let's say in astronomy, for example, some of the areas that the Kavli Prize recognizes and very important areas, or that the Carnegie Institute supports, it's an observational science. Okay, in the same way that evolution is an observational science, and therefore it's harder to prove things with the same absolute certainty as you can in a particle accelerator or in my test tube. Okay, so we don't think we live in a world of tremendous uncertainty, actually. Okay, but when it comes to things that matter in the real world, okay, actually where all variables can't be controlled, then that becomes a real issue. And I think that, for example, in the issues, the debate about climate change I mention that because I think it's a perfect example of scientific concepts that can be applied, but in a real-world situation where, by definition, we can't do an experiment. We think we're doing an experiment, actually, perhaps unintentionally, but in the end, we can only make the most educated guesses. So we have to make good decisions based on that uncertainty.
You and I spoke about this in this environment, and Michael asks the question, and 18 people would like to know the answer, your thoughts on this as well, which is, what do you think about the state of science funding in the United States right now?
Okay, well, actually, thank you for Michael and all the others for providing the opportunity to answer that. At one level, I'm encouraged, okay, because our Congress has stood up and once again reaffirmed its long-term position supporting and sustaining medical research in particular, but science generally. And I think we should all be really grateful for that. That's just a huge thing, and it's not to be taken for granted. Having said that, I think that even before this year's debate, there's been a sea change during my life as a scientist, which has, despite best efforts, science is a productive discipline. It always attracts young people. As a result, we propagate. We train PhD and MD students. We have a generation time, if you want to think of us as a, hopefully not a microbial infection, but more as an opportunistic parasite. Because we do cost money, but we do add a lot, okay? Probably more than what we cost, okay? We generate new scientists with generation times of 5 years, maybe, roughly, okay? So we expand and we expand and we expand. Now, we can't expect to grow to the point where supporting science is 99.99% of our gross domestic product, obviously. So there's a process that is unavoidable, in which science growing faster than the gross domestic product, which has a doubling time perhaps of 10 or 15 years, even in good times, we become much more prevalent. So we've grown and grown and grown relative to the society that supports us. And obviously that has to stop and reach a steady state at some point. When I was a young scientist and applied for my first NIH grant in 1978, that year I actually checked these numbers. That year we had a 45% chance of getting funded. Now it's about 15% on a good day. Okay?
45 to 15%.
Right, okay. The size of my grant, when it was, which sounded like a lot at the time, wouldn't sound like so much today. But if I use the NIH's own biomedical cost inflation, whatever index, to inflate it up to the current value, would be about $400,000, maybe $450,000. So to summarize, 27-year-old punk kid has no preliminary data, goes to work in a good department, has people write good letters about him, whatever, okay? Has no preliminary data, wants to do something completely radical, writes it down on a piece of paper, does it before he even takes his job, okay, when he's a postdoc, 45% chance of getting funded, and has the equivalent of 2.5 current standard issue basic science NIH grants today. Wow, that's huge. And then turns around within a year, based on a few measly results, and gets a second NIH grant. And suddenly, that is what science was like. That is when science truly prospers. Now, I'm not blaming the NIH for that, because, as I said, we're growing, and the NIH can't grow infinitely. But we all can cooperate in some way or other to make the situation better. And I actually think that all the parties in the equation need to up their game. Universities, we just grow without thinking about it, okay? You want to build a new building? That's great. We put it on NIH overhead, okay? I think there's been a lot of talk about reducing overhead. I think actually that's destructive. But I think that it's also destructive when our medical schools and research institutions expand essentially as real estate on spec. And that's been going on for the last 20 years. And so as a result, without a lot of thought, we expand, and the funding has been there ex post facto to sustain it. We have to do some hard thinking about the size of our faculty, and we have to do some hard, and we also, a lot of you guys probably don't know this. Do you know that a typical professor doing medical research in a medical school gets very little of his or her salary from the school, despite the high tuition? Did you know that? Okay. Well, it's understandable because actually we couldn't afford it, otherwise. But you have to ask the question, maybe we need a different model. Maybe we should be having more skin in the game as universities and actually funding what we do. Okay? Not 100%, maybe even just a few percent. Maybe even just taking more responsibility. I know the NIH has asked medical schools and universities to do that, and I know our deans don't like that, but actually, many of us think that that's important. So we need, and then we have the professoriate. We have to up our game too. It's actually not reasonable to double the scientific, the number of hungry mouths consuming for productive reasons. Believe me, if we could continue to grow exponentially, society, I believe, would continue to benefit exponentially, and the American entrepreneurial economy would continue to benefit. But we have to find a way to be very productive within a smaller envelope. So we have to do that. The professors, we have to maybe think harder when we take a graduate student. Maybe create career paths that are non-professorial to a greater degree than we have. And all of these discussions are going on, but in reality, we haven't made that adjustment yet. And now I come to the government, okay? I mean, the government needs to, if it doesn't come up with if it doesn't do, for example, what the German and the Swiss governments do, which is have formulas for sustained budgets and funding over 5-year or longer cycles, it's going to be problematic. And in particular, I would point my finger at the NIH over the last decade or two, especially the last decade, as having incrementally moved towards more big projects that are top-down. And I know that if someone from the NIH were here, they'd say, well, it's only this percent or that percent. And I'm sure they're right at some level, but the fact of the matter is, from where we sit, that as individual investigators, that we're feeling under enormous pressure to do research that's more applied, and we need applied research. And one of the great things that's happened in the last 10 years is we've had more focus on translational research. That pendulum needed to move. Okay? But now that momentum is so high that I think it needs to be corrected a little bit. And I think that, and I know the NIH is starting to make moves in this direction, and they're really good ones, and they're really welcome. But I think we need to have much more in the way of going back to what made American science great and made the NIH investigators great, which is the guy in the lab or the gal who have a great idea, and it didn't have to be all proven out first, and you take a risk, and you give them the opportunity, and it's investigator-initiated, rather than some bureaucrat saying, we need to have a $20 million project to do this, getting advised probably by an in-crowd of people who then become the people that get those grants. You know? If that were happening and it was real estate that was being sold, you know what we'd call it, okay?
Brooklyn Bridge in Arizona.
Something like that, okay?
That resonate with anybody? A very thoughtful, very interesting answer.
But I don't mean to put my finger just on the NIH, because I really don't.
Well, no,
we all do that.
You pushed everybody there.
But the academics, we all do that. Oh, the NIH. They've got their bit, but we got our bit, and we're not doing our job.
In a few minutes, I'm going to open it up from the questions that you've brought to us via Pigeonhole to some who might want to go to the mic. But before I do that, I want to go to the question here that has the most votes.
Oh, my goodness.
The most votes, which is, what effect does your mood have on how your brain communicates?
Well, me, it's really profound.
Well, do tell.
You mean in a general way?
I'm wondering whether I can't interpret the question unless you're in the room who authored the question.
Okay, so I'm not really a neuroscientist. I mean, it's a really good question, and I know from my own personal experience, like I came in here, and frankly, I wasn't in the best mood. I had to answer all these questions and stuff like that. So I asked for a cup of coffee, okay? And Natasha will remember that she got me a cup of coffee and my mood improved, okay? So I guess you might say that caffeine, which actually is a mimic of a neurotransmitter, modulated my limbic system in such a way as to improve my cognitive ability. But, so I could give you an answer like that, but I think we all know that if you're in a good mood, it's one thing, and if you're a bad mood, another. I happen to be particularly a moody person, and any one day I can go from very high to very low, okay? My wife tells me that's not a good thing. But anyway, but I think she loves me for it anyway. She's also a scientist, a neuroscientist, so she
understands.
She does the same thing, and I can tolerate it. So I don't know. I'm not sure what the answer to that is, but there is a scientific answer, and actually, even though I'm not a neuroscientist, as a health, I received this Kavli award in neuroscience. God knows why. But it's actually the elimination thing, okay? Because you eliminate something that's really broad. Actually, was it neuroscience? Yeah. Was it cell biology? Yeah. Call it what you want, okay? The point is that I think what's going on at a neuroscience level is that every bit of your cortex, so just to get to a bit of science, so you have a little bit of cortex, we have a bunch of synapses, and they're talking to each other, and they're firing, they light up in an fMRI. You've all seen that, okay? Did you know that every little bit of the cortex is bathed in mood? Okay? So there's other parts of the deep brain, the amphibian brain, the primitive brain, the brain that actually determines what we do. Okay? The political brain that literally drips serotonin, the mood neurotransmitter, dopamine, the reward neurotransmitter, and several others. Okay? Acetylcholine, the attention neurotransmitter. It drips them like a little burette on these cortices. And the cortices remember what it was dripped with, essentially. This is my neuroscience view. They remember that pattern, and that's how they encode emotion. Okay? So every memory that you have, in a way, has an emotional overtone. And so I think, actually, that the connection between, at a neurological level, between mood and what we would think of the ethereal neural circuit is deeply profound, and it's not actually that well understood. It's a very important problem in neuroscience.
We're going to take now some questions from the floor, so if you'd like to pose a question in the few minutes remaining, make your way to one of the microphones. I'll ask you to be brief with your question and succinct so we can get as many in as possible in the few minutes remaining. Sir, tell us who you are and go for it.
You've actually started discussing this. Is there a backup system? I mean, is this 100%? Does every emission get to the right receptacle, or is it a probability thing? And also, the concept of disruption of the process. You talked about genetics and predisposition. How about like things like stress and cortisol and things like that? So.
Okay, well, on the second question, they're both really good questions. And the second one, I'm a dropout of medical school, so I'm going to take a pass on that one. Okay, on the first one, it's actually really, I think, an interesting question. Does the cell get it right the first time? Okay, or does it have another go at it and correct its mistakes? Okay, and actually, the answer is in the processes I study, it gets it right mostly, but it has a way to correct its mistakes. Okay, and actually, to generalize a little bit, in biology, okay, it's always that way. We have to have a very low error environment, starting with our genome. Our DNA has all the time is accumulating mutations for one or another reason, and we've got to get rid of them. It makes mistakes when the DNA gets copied. When a protein gets made, occasionally, not so occasionally, the wrong component amino acid gets incorporated, and it needs to be that protein needs to be removed. And in the case of vesicles, actually, you usually get multiple goes at it. And actually, one of my aha moments, okay, which is still not fully accepted by the scientific community, but actually the discovery that I am the most proud of, okay, that most people would say is probably still more a mistake than a discovery, but I like it, okay, is exactly that. Do you remember I showed you the electron micrograph of the Golgi apparatus, and it had these layers? It looked like a series of flattened pancakes, one on top of the other, and the vesicle was going from one to the next. It turns out, increasingly we recognize that is a distillation tower. It's a process where the vesicle does the first one, and if it gets it wrong, what it delivered wrong goes back. And then what it delivered right goes on. And so when you do that step, that transfer over and over and over again, it's exactly the same principle that refines oil into gasoline in these big towers that you see nastily in Secaucus, New Jersey, outside of New York, okay? That's exactly the same physics, okay, I believe. And I remember late at night in my office at Stanford when in 1980, that idea hit me for the first time as an accumulation of a number of facts. That was actually my best aha moment ever. So far, I'm still the only person that's aha-ing it. But I think it embodies your question, okay? So thank you. Thank you for a great question.
Thank you.
Hi, thank you for being here today. I wanted to ask
Tell
you...
us your name, please.
Oh, sure. Hi, my name is Alicia Suchisitl. I'm a senior in bioengineering at George Mason University.
Great. Thank you for coming tonight.
I'm really excited to be here. Thanks. I wanted to ask you how might neuroplasticity and learning be affected by the rate of membrane fusion and vesicle transport?
What's the word neuroplasticity?
Neuroplasticity.
Okay. So I'm going to define neuroplasticity briefly for other people. Okay? So what neuroplasticity really means is it's fancy words for memory. Okay? So the basic principle of memory is that, when the nerve cells communicate with each other, they do it by these synapses where the neurotransmitters are released. I think I've mentioned that several times. If you use a synapse a lot, it gets stronger. That means that if you have a certain amount of electricity coming in one nerve, you get more out of the other. So that synapse is said to be stronger. Okay? That's the basic principle of memory, and it was discovered by my colleague Eric Kandel, for which he received the Nobel Prize in the year 2000. Okay? Very fundamental. Okay? Now, underlying that, plasticity means if you use it, it gets stronger, so it was plastic. If you don't use it, guess what? You'll lose it. Okay? So, very important. Okay? Also very important in neurodegenerative disease. Use it or lose it. Now, the vesicles, it turns out there are vesicles on both sides of the synapse. So I've been lying to you. By omission, not commission. On the presynaptic side are the synaptic vesicles that have the neurotransmitters. Now, more recently, it's been discovered on the postsynaptic side, where the neurotransmitter goes. On the receiving cell, there are the receptors for the neurotransmitters. But what's more recently discovered is they're also in synaptic vesicles. Similar, but they have the receptors for the neurotransmitter in them. And when you use the synapse more and more, these receptors on the postsynaptic side are delivered more and more to the surface, so that when you have a certain signal coming in, a certain amount of neurotransmitter released, you get more output on the other cell. That is the fundamental event, probably a fundamental event, but I think the fundamental event underlying memory. So actually, and it happens in the short term plasticity, where this is regulated over minutes, but it also, when this happens enough, the synapse sends signals back to the cell body that then render it permanent through genomic changes in the cell body and the nucleus. Okay? But that's the basic principle of memory, and this actually, regulating this fusion process quite directly underlies that. That is a really good question.
Thank you.
From the scientist's point of view, okay, that's really great. That's a young scientist question. That's really great.
Bravo. We're running over, and I'm mindful of that, but we also have 3 questions, and let's try to get them quickly in, and quick responses so that we can get you all on.
I'm sorry,
the rest
I'm
of
guilty for the long response.
You know what? I think we could sit here and listen to you all night and into tomorrow.
Well, thank you.
I mean it. I think he could be the John Oliver of science if he ever wants, right? I mean, you're clear, you're funny, you're brilliant, you're an amazing communicator.
Well, we better stop now.
Yeah. That's your question.
Hi, I'm David Mogg. I taught physical science to high school students for 20 years, and they used to get extra credit for coming to these events. But I'm now retired, and I have a mom who, just like you, never figured out why I didn't get a medical degree.
I guarantee you don't have a mom like me. Because I got to tell you, a month after the Nobel Prize, she calls me up and she says, I read that you can get 2. And I'm not making that up. So what's your question? Sorry.
My hero, Linus Pauling. Anyhow, how well preserved? My question is, I get it that all cells use the same mechanism with the vesicles and those proteins, the snare proteins.
Yes.
How well preserved is the sequencing of the snare proteins?
Okay.
Are they identical in every kind of cell, or is there variability?
So, okay, let me tell you one that will really answer that directly. So it's a really good question. So if you take a snare that mediates a particular transport step in a human, and you put it into a yeast cell, you can replace the yeast gene that mediates the corresponding step. So it not only fits in, but it partners all the other yeast proteins to get the job done. Now, that means that this code in the snares for membrane fusion is as fundamentally conserved as the genetic code. Okay? And it ought to be, because the genetic code, its purpose is to make proteins. The DNA encodes an RNA, encodes a protein. Okay? The purpose of this code is to put the protein in the right place. So actually, what's the point of making a protein if you don't put it in the right place? Getting it in the right place is the point of the entire genetic code. Okay? Good
Next.
question.
Thank you.
Hello, my name is John Putziger. I'm a McLean High School senior. I want to go for chemistry in college. So I want to take vesicle point of view from a chemical standpoint.
Yeah.
Why were the basic parts of vesicles made out of lipids instead of other polar compounds?
Oh, and now you're asking me a why rather than a how. Okay, now, no scientist will ever answer a why as compared to a how. Okay, but since I'm not a scientist tonight, I can answer that. Okay. No, I mean, I think that no one knows, of course. I mean, I don't think anybody can possibly tell you with a straight face exactly why things evolved the way that they did, and I'm not going to pretend to do that. But I will say that phospholipids, in particular, are ideally suited to form membranes. And the reason is that they have a fatty tail, which is just like salad oil. Okay, literally, it's made from salad oil. Salad oil is a diglyceride, okay, or olive oil. It's two fatty acids, and it has a head group attached to it chemically, which, as you know, is from your high school chemistry work. AP? Good. So you know this for sure, okay? So, and that part is called the head group, and it might as well be in the water because it's a very polar soluble molecule. So this molecule is a molecule in need of a psychiatrist. It doesn't know where to go, okay? Because if you had salad dressing, how many of you have shaken up salad dressing, oil and vinegar, right? What happens if you just let it stand after a while? It's very frustrating, right? Separates into oil and vinegar, right? Vinegar is basically water. So if you take the head group of your phospholipid, shake it up in salad dressing, it ends up in the water. If you take the hydrophobic part, the tail of the fatty part, you shake it up in salad dressing. Where does it end up? In the oil. This molecule can't decide which way to go. Oil or water? So it goes right in the middle. You know, it comes up with a compromise, okay? Now, which, by the way, no neurotic person ever does. And as a result, it has to form a surface that segregates from the water, where its head is exposed to the water, and it pairs with an equal and opposite surface to create its own environment. That's called the membrane. And I think that molecules like that are probably created naturally, okay, through polymerization molecules with carbon, and in an oxygen environment, okay, they will get oxidized and charged. So I think it probably requires some oxidation. And I don't think it could have occurred very well before oxygen. So I have a little bit of a problem understanding how it evolved from that point of view, because we know life evolved before oxygen, because we know oxygen was produced by plants. So the why, have no freaking idea. The how, I can answer that for you pretty well. Okay. I hope that helps. Thank you.
Thank you.
Good luck with your
And good luck.
studies. And I hope we see you at Yale next year, because we need chemists at Yale.
I think you just got an invitation to apply to Yale, so...
Now he's back, okay.
Actually, to be fair, I was talking to the Caltech baseball coach. So.
All right. All right.
Your initial dichotomy interested me. Why are those pins not anatomy? I mean, you said that it was anatomy, not specificity, and it's something about scale. But why are those pins, which bring the vesicles together, not part of the anatomy?
Okay, so I would say actually maybe it's a semantic issue to some extent. It's a good point. They are anatomy in the sense that in the ultimate version of anatomy is visualization. And visualizing those pins was actually done by a colleague of mine, Reinhard Jahn, in Germany, and Axel Brunger now at Stanford. And they did that by X-ray crystallography, which is a visualization tool that, it's not a microscope, but it's of the ilk of a microscope. Okay? So in a strict sense, that was anatomy. Okay? On the other hand, the function, which is what, as a biologist, which is what I worry about, was on the basis of chemistry. Those two parts of that structure that was so beautiful, that pin, it actually assembles from disassembled parts spontaneously as a result of protein folding, And that represents the interface of chemistry and physics. And it is not anatomy. Now, anatomy, and what I meant in that distinction really is that the anatomy at the level of histology is not determining the outcome. It's rather that the anatomy at the level that you would see it in a light microscope is the consequence of the underlying chemical rules. That was really what I was driving at. But you're perfectly right to say that the end result of our understanding is you could think of as anatomical in the sense that any chemical structure is a form of anatomy.
Thank you very much. I will close with this question, and it's something that you and I kicked around when we first chatted. And I said, so what do you want me to ask you? And he said, a good last question would be this. If you could live another 100 years, what would you like to get your arms around? If you could live another 100 years, what would you like to get your arms around?
You know, that's a real good question. Yeah, I don't know. I could go on for a very, very long time with that.
Don't do that.
Yeah, I won't. It's a serious question. And my answer is always the same, okay? Which is I could give you an answer, but you should not listen to it. Okay? In other words, don't take advice from old scientists. Okay? Because if I could answer that question, I mean, by definition, the person who can reasonably ask and productively ask a question like that is a person of the milieu currently. Okay? I am a creation of the milieu that I described to you. So if you asked me any question that was important in 1980, I could probably do as good a job or better than most people in formulating it in a good way. But today, I would not trust myself. I would rather trust any of the young people in the room, as long as they don't take advice from people like me. Okay?
Fair enough. Your mother asked you if you could win a second Nobel Prize. I'll ask you if you could win a second Kavli Prize. We'll let you keep working on that. I would like to thank you for the incredible things you've done, the passion that you've brought, the dedication that you've brought, the discovery, the lamp that you have lit. And thank you for a fascinating presentation and discussion this evening. Please join me in thanking James Rothman.
And I would just like once again to thank everybody for staying for a long evening, and I think we should thank Mr. Cesno as well for making himself available and doing such an incredible job.
My pleasure. Thank you. Thank you all.
And I have to say, I feel like I made a new friend.
I do too.
Yeah. So